right, well, thank you all for coming. My name is Bill Rihl. I'm from Berkeley National Laboratory, and I'll be talking about the KBase project a little bit today and about the KBase narrative in particular. So what is KBase? Has anybody heard of KBase, first of all? I'm curious. Oh, great, there are people out there. And not just people I know from Berkeley. So KBase is a platform that aims to enable collaboratively developed, data-driven, formally testable models of biological behavior, and specifically we're targeting within an environmental context. And so we're part of the Department of Energy, so we're looking at our main biological targets right now are microbes and microbial communities taken from environmental samples, as well as plants and plant life and how that whole biosphere interacts with each other. So the goal of KBase is also to be able to build all of these models and analyze this data and to provide provenance and reproducibility for all of that, which are two great terms that I keep hearing over the course of this conference, which is very exciting. And to, one of our big goals is really to bring together both computational scientists, like everybody here, and also experimental biologists that may or may not have the coding background that computationalists have. We really want to bring everyone together in a way that in a ecosystem that can all work together to produce good biology. So we do this by, our goal here is to empower users in a broad system that will incorporate all of the data that they want to use, store it locally, or store it, on the KBase system in a way that it can be provenanced and kept private as well. So I guess going from the upper left side, I don't really have a pointer here. A user would be able to either upload data of their own or download it from a public data store that we've collected from the public databases. Introduce metadata on top of all this, use their own particular contributions, integrate this together and store it in a private workspace system that we're calling, so that they would all have access to all of the data that's there and present. Use the KBase system itself and a number of API and method calls that are already available and continually growing to do analyses on these data objects. And in the end, produce comparative analyses from these data, visualize the outputs, and really generate hypotheses and predictive models of how the different things would really act in nature. So I mentioned a little bit before this workspace, this private data storage idea. So to go into a little bit more detail with that, for example, I would have, I guess that's not terribly visible. So a workspace is a way to, so the KBase workspace takes together all of the different data objects. We type them out specifically, so you would have a specific genome typed object, for example, or a metagenome typed object, or expression data typed object, and so forth. Store them within the KBase system so that they may maintain accessibility to you or to your collaborators in a way that you can share with each other, and also have that type of accessibility to the different methods and software services we have. And also we have a way to visualize each of the data components. For example, this one here is a flux balance analysis model or a metabolic model. Clicking on that just within the system itself, we can dig into it and see what's there, what it's about. So once a user would have all of their data accessible within their workspace, we have hundreds of functions and different API calls that we generated both internally as part of the KBase team, and also externally we've brought in a number of open source third-party packages to be accessible to all of this, including Chime and some of the BioPython type things. So, and all of these range from different genome assembly and annotation to metabolic model building, metagenome analysis, GWAS systems, and so on and so forth. But really the way that we envision having people access these and work with them is through the KBase narrative. So the narrative is intended to be our major graphical interface and into the KBase system. And this is built on top of the IPython notebook. And it's meant to be both a teaching and a publishing interface for the KBase platform. So it's useful to So we've had professors right now being able to use it to show their students how to do different biological and bioinformatic tasks. But we also, in the future, and we're not quite there yet, but we want to be able to publish these workflows in an immutable way. So it's almost like having a paper that you would publish in a standard journal, but you could publish your narrative and your workflow online and the data that goes with it and have that shareable by the community. The next goal for the narrative, as I mentioned before, is that this should be an easy-to-use interface to the KBase system, not just for coders and people that are comfortable programming and doing the grungy work of coding in bioinformatics, but also for non-coders, people who might be data generators, experimentalists that want to be able to work with all this, but may be a little bit scared off by having to learn how to code. Some more detail. This is intended to be language agnostic. Currently, we have support for Python, R, JavaScript, and our own KBase scripts. And we have upcoming support, hopefully, for Perl, Ruby, and Julia, which are also up and coming in the bioinformatics field. And really, this is an interface that we don't just allow collaboration, but we promote collaboration between the coding types of users and the experimentalist users. So how do we do that? So we KBase enables ad hoc kinds of workflows. So if we have a single user doing a single pretty linear workflow where you might drag in data that you would search for data, particular data, say sets of genomes and so on from our central data store, public data stores, or you might want to upload your own data. Maybe you have, for example, a series of reads that you've made, sequence reads that you've made from an environmental isolate. Bring that into your workspace, analyze it, perhaps assemble these reads together using various algorithms into a genome, and be able to visualize that genome, decide do you like it, go on further and build, for example, a metabolic model out of it, and try to predict how that model would behave in different environments, and eventually go on and publish this whole thing. But we also have our other goal is also to be able to collaborate with other researchers that might be doing the same things. And the main way that we intend to do this is by sharing these different data objects and the different narratives. So another user might come in and that you would collaborate with, draw data from your workspace that you've been working on, integrate it into their own analyses, share back and forth, work on the same narratives together, and publish either together or separately, and also incorporate different visualizations and so on. So I'll go through the narrative itself now and how that looks like and how we intend that to behave. As you can see, it's built pretty heavily on top of the IPython notebook. The right side of the panel there is the modified version of the notebook itself, and we also have accessors to the data and some click button services and widgets that plop into the screen and plop into the notebook. There's more detail about that. Up in the upper left, there's the data panel itself, and this is a mini view of your current workspace that you're working on that has all of the data that you've either accumulated or built based on analyses. And we can dive in deep and see more details into that. So for example, here, if I click on this guy, which is a genome, and man, I'm really sorry about the font size. That didn't really work out. But we could see some more details about what that genome is. Here, this is a Pseudomonas tutseri RCH2 genome, and this panel here will tell you all the different metadata about it, what typed object it is, when it was uploaded, the most recent version that was saved of it, lets you toggle between different versions if there have been different analyses that have been done that may not have the right parameters that you want to use. You might want to go back and change it around some, and so on and so forth. Below that data panel is our services panel, which has a simple introductory click button view of many different KBase services and KBase method calls, and these are all written right in Python and are used to extend the IPython itself to become the narrative. For example, this one here that I show on the screen, this one is very simple. Just, I want to grab a genome out of my workspace and I want to view some details about it. So a very small script is written that gets compiled into the narrative system. And it's really just a bit of Python code that's annotated to say, this function is view genome. It takes in a genome-typed object as an input, and it outputs a genome itself as the output. And by just using a few simple annotations, it will produce this data widget here, which will auto-populate with all the genomes that are present in your workspace, for example. The user would then just click run, and the output would pop up. And this output box here is a JavaScript widget. So we're kind of integrating JavaScript into the IPython notebook, maybe a little heavier than most people would like, but we're getting there. And after seeing John's lightning talk yesterday, maybe there's a way to transfer that into just pure Python instead, which is pretty exciting. But in the end, this particular widget will just show some more details about what the genome is that the user is interested in. And this is a great, even now using JavaScript is a nice way for visualization because there's a lot of different packages that are freely available, including D3 and SVG-based ones, and upcoming and improving support for HTML5 and Canvas and all of that. So our goals with this particular function panel is that it should be easily extensible by any developer who wants to get involved with it. And it's just based on IPython traitelets and annotations. And it can go, these function inputs and outputs appear right onto the screen and in line with the IPython notebook itself as optionally customizable JavaScript widgets. And I showed a very simple example of just grabbing a genome and displaying it on the screen, but they can be rather complex and invoke complicated functionality. But this eases the way in for non-coders. So an example of a more complicated system would be to take an annotated genome and build a metabolic model out of it, and then run further analyses to test that model against known environments, see that it actually behaves as though we know that it should behave in that particular environment. And this can all be wrapped up into these little widgets. And currently this is all only in Python, but other languages are on the way if people are interested in using those. But since this is the IPython notebook, we also have, for more advanced users that are more interested in doing the coding analysis themselves, writing scripts, that's certainly possible. All of the KBase scripts and API calls are available as part of the narrative. For example, this one here will just, this reasonably small little block of code will search for all instances of Bacillus subtilis, grab them from public data stores, import them into your workspace, build a metabolic model out of them, and test them all in just, I don't know, a few lines of code there. And again, since it's not, this is the IPython notebook, it's not just limited to using KBase, but any kind of other numerical analysis and functionality. So a little bit about the details of how this all works. The term Docker has been bandied around this conference a little bit, and so what we've done is similar to what other folks have done, which is build our narrative within a Docker container. And are folks, anybody here familiar with Docker? Raise hands. Okay, for those who are not, a Docker container is a self-contained Linux environment that sits within a virtual machine. So it's not as heavyweight as a virtual machine itself. Docker containers are able to sandbox everything within themselves, so they don't necessarily compete with anything outside of their environment. And it gives some great advantages. They're reasonably small to build and to store. They're very, very fast to start up. As soon as a user goes to the page and clicks go, then they have their own sandboxed narrative instance made specifically for them. And they can swap out and shut down pretty easily as well. And within this container, we have our own Python virtual environment that contains the IPython notebook, on which we've built on top of with the different KBase widgets and the KBase client libraries, and these get exported straight to the client. And as well as everything living in the back end on the kernel are the KBase IPython modules and the client libraries that talk to the KBase servers and run the different service calls. So we are really just getting started with all this. This is still a very early project, but there is a demo version available, an early demo version, up at KBase.us. We are a fully open source project, so check us out at GitHub. And if you are curious about it at all or have any concerns or want to see where we're going with it, check us out at KBase.us. So our future plans are to really get improved support for R and other languages. All of that's built into Python right now, but our goal is really to have a user run their jobs, run their different code cells, and store all the output within their workspace so it keeps that same provenance model that everything else has. We want to involve management of long-running jobs. Many of these analyses, as I'm sure you all know, can take a very long time to run. If I have a couple terabytes of metagenomic data, it's not something that I can click on a code cell and wait 30 seconds for it to pop up. So we're improving our management of that. And we always have more and more visualizations to build for every different data type. And we're expanding our GUI to make it accessible to everybody that wants to use it. And of course, sharing and publishing not just the user-created narratives, but also widgets, and having people from the community come in and build their own function calls and visualization widgets. And eventually, we would like to get to the point that a narrative can reference and pull in pieces of other narratives to be able to swap out different workflows in a pipeline kind of way, almost. So a little bit about the KBase team. We're a pretty large team. There's about 80 of us spread across 4 national labs, Berkeley, Brookhaven, Argonne, and Oak Ridge. We also have strong collaboration with Cold Spring Harbor Laboratory and a number of colleges and a number of university professors we work with. And again, I would just like to mention, come check us out at kbase.us and be happy to take any questions.